Hello guys, how are you? I just wanted to make a video. This drawing is gonna be too bad, but hopefully you would learn, if you don't know it, the basics of the pathophysiology. Um, I think I post I think the same thing that I'm going to explain here, but I think it's not pretty comprehensive if you write that down. I think that it should be explained through, I don't know, through examples or through drawing, if you want to make yourself clear in this point. So talking a little about the pathophysiology of the disease, first of all, we're going to have that COPD is divided into chronic bronchitis and emphysema. There are other things too, for example, asthma and COPD disease. That is another type of disease. It is not asthma, it's another subtype. And there are many others, but I think those are the most common and the most important ones. I'm going to start with chronic bronchitis. I'm going to do a drawing here. I'm going to draw the airway first. This drawing is going to be pretty bad, but hopefully you will see here that you have the here you have the trachea here, and then the trachea further divided into bronchi, main bronchi, and the other subtypes. And here you have, for example, a bronchi, then the terminal bronchiole that is right here. There's the respiratory bronchiole, and then the alveolar sacs. So now what we're going to do is explain that inside the alveolar sacs you will have inflammatory cells, neutrophils, macrophages, and stuff, and many others. But basically neutrophils and macrophages. Just stay with me there. I don't see the point, honestly. I've always been like this, of learning a lot of stuff, like a lot of different molecules and stuff, if we don't see the principal picture. So let's start with this picture that is like the most basic stuff, and then we can add the inflammatory markers, the inflammatory cells, the immunity, and all that stuff related to the COPD. I think that in this way we can all be in the same level. So right now, well, we have the inflammatory cells, and for example, here is the neutrophil. The neutrophil, it produces a lot of elastase, for example, but also produce metalloproteinases in some sort, and produces also trypsin. And this neutrophil is going to produce these inflammatory markers that I just mentioned, because there are a lot of stuff in the environment that enters the airway, and produces here an overproduction of this inflammatory marker, this inflammation in the alveoli. One example is the is smoking. Chronic bronchitis is well known because of smokers, but there are other examples too. That people that used to cooking wood many years ago, they are also with chronic bronchitis. I've seen in the hospital a lot of people with this disease secondary to cooking in wood. So smoking is not just the main thing here. So this production of inflammatory markers gonna further produce a thickening in this wall. So if we extrapolate this wall, we can see that the wall will have many levels, but basically three. We will have an epithelium. We will have a space that is composed of submucosal glands, basically, submucosal glands, and a space that is composed of perichondrium that forms the trachea. Okay, that's pericardium, right? So here, the submucosal glands. Here, no, easy. So here, the submucosal glands are gonna be thicker, right? Gonna be thicker, and that will produce that will produce that this wall is going to be thicker too. Like this space is thicker. There are more submucosal glands here that produce an increase in mucus, because of the inflammation that we just talked. And this thickness of the wall is going to be represented in something that is named the rate index, something that is named, that is pretty famous actually, that is named the rate index. Rate index. So the rate index is when this, well, as soon as I know, the rate index is when this space of submucosal glands is increased in more than the 50% of the total of the wall. So what is the problem that we have here? I'm gonna write here problems, right? No, in black. Problems. First of all, what we have is a thicker wall. First of all, we have a thicker wall that causes two main things. First of all, air trapping. Air trapping. And second of all, we're going to have a problem with the diffusion, right? With the diffusion of gases. Why is that? First, let's start by talking about air trapping. Okay, if we have this wall that is thicker, in the bronchioles, the air that will be here will be pretty hard to get out of the alveoli. And if it cannot get out of the alveoli, it's going to stay in the alveolar side. So if we do an spirometry, and a spirometry is an exam where the patient has to get the air out of the body and stuff. And if you measure the parameters of that spirometry, you will have an increase in the TLC. First of all, TLC, total lung capacity. That is the amount of volume that will be in the respiratory system. And most importantly, it will have an increase in the residual volume. Residual volume is the volume of air that stays after the expiration, right? Okay. Diffusion. So if we have the diffusion, you remember? So what is the problem in the diffusion? So if we have a thicker bronchi, or if we have a thicker bronchiole, the diffusion of the oxygen, let's draw the oxygen blue, the diffusion of the oxygen is going to be more difficult, right? So this would cause an hypoxia first, that will cause an hypoxemia after. Why is a hypoxemia? Because here below the alveoli, you would have here the pulmonary vein that came from the right atrium of the heart. This pulmonary vein is gonna divide into capillaries that will touch capillaries of the pulmonary artery, in order to transform to a pulmonary artery, right? So here is a diffusion of the gases. Here's what happened, or where the diffusion of the gases happens. So if you have less oxygen, that causes an hypoxia or decrease oxygen inside the alveoli, you will have a decrease in the oxygen that passes to the blood. So you will have then an hypoxia and an hypoxemia. Hypoxemia, that is the decreased oxygen inside the blood vessel, basically, inside the blood vessel. But what happened with the CO2? What happened with the CO2? That you know that the CO2 has to pass through the capillary to the alveoli. It's gonna do the totally opposite of the oxygen, to go from the alveoli to out of the body. You have to get rid of the CO2 that produces your body, your muscles and stuff. And that CO2 cannot go out. Why? Because there are thicker walls. So you would have an increase also in the CO2 inside the pulmonary artery, and this is named hypercapnia, right? So this hypercapnia is going to cause a respiratory acidosis in the patient, right? A respiratory acidosis, that is a decrease in the pH of the blood in the patient, that will cause other changes in the body, and an increase in the reabsorption of bicarbonate in the kidneys, for example. Yeah, and a cascade of things inside the body that we can talk a little later about it. But this is like a picture, purely to draw a picture of everything that happens in chronic bronchitis, that it is diagnosed clinically. But if you are coughing, productive cough, for more than 2 months in 3 different years, or I'm sorry, it is the opposite, the 3 months in 2 different years, you can do the diagnosis of chronic bronchitis, right? So let's erase this to talk right now of emphysema, because here we have COPD. COPD is not just one pathophysiology. That is not correct. Like, let's erase everything. So emphysema. COPD is not just one pathophysiology. There are many pathophysiologies, at least two different ones. So emphysema. Emphysema, it could be drawn as the same. We can start with airway, bronchioles, alveoli, bronchioles again, alveoli, bronchioles again, alveoli, bronchioles, bronchi, and we're done. And here you would have the neutrophils and the macrophages. There are others, but these are the most common inflammatory cells. So let's talk only about this, because we want to set a basic picture of what is happening here. So this is the same thing. There are allergens that came here and that further produce an increased reaction of these inflammatory cells. For example, macrophage produce a lot of metalloproteases. I think that is the most common metalloproteases, and the neutrophils produce elastase and produce a lot of things. But I wanted to talk more about the trypsin. Trypsin, right? Trypsin. I think that trypsin is written like that, right? Antitrypsin, trypsin. No, it's not written like that. I'm sorry. Right. So trypsin. So this production of metalloproteases and trypsin is going to produce a damage in the walls of the lung system. So now this wall is not going to look like this. It's going to look like this, right? Why is that? Because all the barrier has been damaged. So now the problem that we have here is that we have a broken wall, and there are some points that I want to make, right? So first of all, the DLCO. Second of all, the gas diffusion. The gas diffusion and air travel. Let's talk in the same. And then the DLCO, gas diffusion, and the elastic recoil. That is pretty important. So what happened with the DLCO? Well, what we're going to have here is a problem in this wall. because it is broken. So you would think, as I thought years ago, that if it is broken, the gases will pass more freely through the barrier, and that is not right. If you don't have the wall, if you don't have the barrier, you cannot do the gas exchange. So if we draw the capillaries again, you remember pulmonary vein and capillaries, capillaries from the other port, and then pulmonary artery. What we will have here is as the DLCO is damaged, we cannot pass for example, the oxygen from here, to outside the body. So we will have, the CO2 from here to inside the body, to outside the body. So what we will have here is an increase in the CO2 inside the body, but we cannot also pass the oxygen that came from the environment to the alveoli, to the capillary walls, causing an hypoxemia again. So it's the same thing, produce the same thing, it's the same outcome, but by different mechanisms, right? So we will have an hypercapnia, we will have an hypoxia, we will have an hypoxemia, just as the chronic bronchitis, right? So you remember the diffusion, gas diffusion, and air trap. So if we cannot pass these gases, we will have a problem with the gas diffusion, what I just talked. Yeah, right. But also, there is a problem that will cause air trapping, causing an increase also in the residual volume, right? An increase in the TLC, and that we just talked, and that is something like pretty important actually for emphysema, and that is the elastic recoil. So So here in the bronchioles, we will have this recoil, that functioning, keeping the airway open, right? So if you destroy the wall, you will also destroy the elastic recoil, right? You will also destroy, I'm gonna copy that here, as elastic recoil. So you will have a problem in the elastic recoil, right? So when the gases pass through the wall, this wall will collapse. And that is why people with emphysema and COPD, when they expirate, when they exhale air, they do it very slowly, because if they do that very quickly, this elastic recoil that does not exist, the airway will collapse more quickly. So they have to exhale more slowly, right? So that is why this wall collapses and closes, and causing that this air stays here, right? Stays here. That is basically what happens in emphysema, right? So now I'm gonna finish right now. So I'm just going to talk a little about the subtypes of emphysema. If you want, we can pause this video right now, and we can talk in a predilection in about the different types of emphysema, right? So that was the difference of the chronic bronchitis and emphysema. I didn't want this video to be too long, so I will this other video that I'm gonna talk. It will be the different types of emphysema. It's gonna be like pretty quick, differentiating the centroacinar from the panacinar subtype, and what is different and smoker from the genetic causes. That is basically. So I hope you can learn the basics of the concepts here, and you can watch it up.